Hello and welcome. Today, we're going to be mapping out the anatomy of the body's fastest reflex, the vestibulo-ocular reflex. For this, our guide is the fantastic, comprehensive clinical review from Australian Dizziness clinics.
We're going to focus specifically on vestibulo-cerebellar integration in the VOR's famous three-neuron arc. And look, this isn't just any reflex. We're talking about the fastest one in the entire human body. What's so crucial for us as clinicians is that its hardwired design is what gives us those precise clinical signs we need to tell the difference between peripheral and central disorders. So here's our game plan. First, we'll get into the VOR's incredible speed. Then, we'll explore how the cerebellum acts as its personal calibrator. After that, we'll actually trace the three-neuron superhighway itself. And finally, we'll look at what happens when these circuits break down, turning pure anatomy into a powerful diagnostic tool.
All right, first up, a reflex that is truly built for speed. The performance specs of the vestibulo-ocular reflex, or VOR, are just, well, they're astounding.
Get this. Its latency is somewhere between just 7 and 15 milliseconds. Now, to put that into perspective, visual feedback alone takes around 100 milliseconds. This means the VOR is acting before your brain has even had a chance to process what the eyes are seeing. It's a pre-programmed, hardwired marvel of evolution.
And here's the absolute key for us as clinicians. This speed isn't magic. It's a direct result of its anatomy. It is a simple, hardwired circuit, and understanding that circuit is everything when it comes to localizing lesions. It lets us read a patient's eye movements almost like a circuit diagram.
Okay, but speed is nothing without precision. And that's where the cerebellum comes in. It acts as this sophisticated control center that is constantly fine-tuning and calibrating this high-speed reflex.
Now, what we're looking at here is the specific region responsible for all this fine-tuning, the vestibulo-cerebellum, which you might also know as the flocculonodular lobe. You can see the flocculus and the nodule right here, and believe me, these two structures have very, very distinct jobs. And this really shows that division of labor perfectly. Think of the flocculus as the gain adapter. It's constantly watching for errors and tweaking how much the eye needs to move for any given head movement. The nodulus, though, that's more like the context computer. It's managing things like velocity storage and helping the brain figure out the difference between, say, a head tilt and just moving forward. Two separate jobs, two separate, elegant anatomical modules.
So the big question is, how does the flocculus actually learn? Well, it uses a cellular mechanism called long-term depression. Basically, when the brain detects retinal slip, that's when the image is still moving across the retina when it shouldn't be, it signals an error. This error signal triggers LTD, which physically weakens specific synapses to recalibrate the reflex. It is literally learning at a cellular level.
And this all builds to an absolutely crucial clinical takeaway. A lesion in the flocculus doesn't break the VOR itself. It breaks its ability to adapt. The reflex is still there, but the calibration fails. So your patients won't be able to adjust their VOR gain and will often have impaired smooth pursuit, a really key distinction from other central signs. Okay, so we've seen the calibrator. Now let's trace the actual hardware that the cerebellum is working on, the raw, high-speed circuit itself. Right at the heart of this circuit is a pathway you absolutely have to know, the medial longitudinal fasciculus, or MLF. As our source material puts it, it truly is the anatomical backbone of the VOR. It's the information superhighway connecting all the key players.
So let's trace this three-neuron arc together. First, the hair cells fire off a signal, which travels to the vestibular nuclei in the brainstem. That's neuron 1. Second, those nuclei send a signal across the midline to the abducens nucleus on the opposite side. That's neuron 2. And third, the abducens nucleus does two things at once. It drives the lateral rectus muscle of one eye, and it sends a signal up the MLF to the oculomotor nucleus to drive the medial rectus of the other. It's just an incredibly elegant and lightning-fast design.
So what is the MLF, really? Well, it's a densely packed bundle of fibers running right up the brainstem. And it's not just for the VOR, it's absolutely essential for coordinating all sorts of conjugate eye movements, especially vertical gaze. And even for our head-writing reflexes, it's a real workhorse.
Now this is where having this detailed anatomical map becomes so powerful when things go wrong. Because if you understand the circuits, you can look at a patient's eye movements and pinpoint exactly where in the brain the lesion has to be.
So let's play detective for a minute. A patient comes into your clinic with a very specific set of eye movement problems. Your job is to trace those signs back to the anatomical fault line.
So let's look at the classic signs of something called internuclear ophthalmoplegia, or INO. When you ask the patient to look to one side, you notice the eye on the side of the lesion can't adduct. It can't move inward. At the same time, the other eye, which is abducting, shows nystagmus. But, and this is a huge clue, when you ask them to converge their eyes, both of them move inward just fine. What does that tell you?
It tells you that the lesion is located squarely in the medial longitudinal fasciculus. That signal from the abducens nucleus that's supposed to cross over and travel up the MLF is being blocked. This is the absolute archetypal MLF lesion.
Okay, now for another classic pattern. Gaze-evoked nystagmus. Here, the patient looks to the side, but their eyes just can't hold that position. They slowly drift back toward the center, and then they have to make a quick corrective saccade to get back on target. It's like they have a leaky gaze-holding system.
This particular sign localizes to a totally different circuit, the neural integrator. This is a group of neurons whose job is to basically convert a move your eye command into a hold your eye command. When it fails, you get this characteristic drift, which is a classic central sign.
So, let's just quickly summarize the key clinical links we've made today. If you see a problem with VOR calibration, you should be thinking about the flocculus. If you see an INO, it's a direct sign of an MLF lesion. And if you see gaze-evoked nystagmus, that points you straight to a failure of the neural integrator. You see, anatomy is destiny, and in our field, anatomy is diagnosis.
But this leaves us with a really fascinating question to think about. We've traced all these incredibly precise brainstem reflexes, right? So how is it that a tiny lesion way up in the thalamus can cause a patient to have profound postural instability, maybe they can't even stand, even when all of these brainstem reflexes appear to be perfectly intact?
We've traced the VOR arc in the cerebellar modules, so join me in video 9, Vestibulospinal and Ascending Pathways to Cortex, where we will map the lateral and medial vestibulospinal tracts to their exact extensor motor targets, and then trace the ascent of vestibular signals through thalamic islands to the parieto-insular vestibular cortex, explaining why thalamic and cortical lesions can produce dramatic vertical tilts without any peripheral test abnormality. Thank you for listening to the series. To explore more similar topics regarding dizziness, vertigo, and imbalance, and to get clinician and patient resource materials, visit AustralianDizzinessClinics.com. Please subscribe and leave a rating.